Okay. So page number 40, we have done that experiment on explaining osmosis using a thistle funnel. What was the conclusion? The water on the beaker in the beaker was passing through the thistle funnel with the help of a cellophane paper, which was acting like a semi-permeable membrane. And we had some sugar inside the thistle funnel that has not passed into the beaker. That means why? Because semi-permeable membrane, the cellophane paper which we have used, can only allow certain small substances. Only the water molecules can be passed through it, not the bigger molecules like sugar and sand, etc. Clear? So now here is a modified version of experiment 2. Taking a more concentrated sugar solution in the thistle funnel and very dilute or less concentrated sugar solution in the beaker, the result would be the same. That is, the level of the sugar solution in the thistle funnel would rise. This again is because some water from the less concentrated sugar solution in the beaker would move into the more concentrated one. So another experiment was done. Same result we got. Okay, from the lower concentration, it was moved to the higher concentration. Then we have rubber sheet and muslin cloth instead of cellophane as a barrier. Now we have used the cellophane paper here as a semi-permeable or selectively permeable membrane. So if we use a rubber sheet or muslin cloth instead, so what will a rubber sheet do? Rubber, it cannot, just a minute. Yes, rubber cannot pass substances through it. Okay, it's a hard substance. It is not a semi-permeable membrane. It is an impermeable membrane. A rubber sheet is an impermeable membrane. So that's why we can see obviously no change in the level of sugar solution would occur in the first case. The rubber sheet is impermeable and it would not allow the water molecules from the beaker to cross over. Then in the second case, the meshes or the pores of muslin cloth are so large that they would not hold back even the sugar molecules. The muslin cloth and rubber sheet, both they are very impermeable. They are not semi-permeable, they are impermeable. So the sugar molecule, they will not hold back. And the entire sugar solution would flow down to a common level due to gravity. The muslin cloth is therefore freely permeable for sugar solution. Okay, here the rubber sheet is impermeable and the muslin sheet is very freely permeable. It is not semi-permeable also, but it is freely permeable. So if we are taking a muslin cloth, then because of this permeability of muslin cloth, that free permeability, it will allow all this sugar solution to pass through. Like not only the pass through, but it will just break out, and due to gravity, all the sugar solution will be, it will be spreaded into the beaker. So that's why we are not taking any muslin cloth or rubber sheet. Rubber sheet will not allow any substance. A muslin sheet, it is freely permeable. So that's why we are not taking this also. Otherwise, it will let whole this sugar solution pass into the beaker. Now, then the very same experiment is performed by using whisking bag. Now, whisking bag is an artificial bag. It's a semi-permeable membrane. So place a sugar solution in a knotted whisking bag and insert a long glass capillary tube till some of the sugar solution rises into the capillary tube. Tie the mouth of the bag firmly around the capillary tube and support it on a clamp stand. And you can see experiment showing osmosis by using whisking bag containing sugar solution. Figure 4.5. You can see a whisking bag. Okay, a knot is there in the lower side and a knot is there in the upper side too. And they have seen the rising level. So some rising level has been seen in the water level inside the funnel or inside the capillary tube. So we can say two key points in the above experiments. There are two liquids of different concentrations. The sugar solution has a higher concentration of sugar molecules, whereas there may be fewer or no sugar molecules in the beaker. It is very important to understand the concentration can also be visualized from the side of the solvent. Okay, then the last line we will read. Thus, there are two regions of different concentrations, tonicity, which you may express in the following ways. two regions of different concentrations of water molecules, two regions of different concentrations of sugar molecules. Now you know in the beaker we took some water with sugar inside it, but the sugar was dissolved. Then in the thistle funnel we took some concentrated amount of sugar molecules. So we are taking two different concentrations here, right? Sugar is of higher concentration and water is of lower concentration. So that's why here we have here we got this conclusion. Two regions of different concentrations of water molecules we have, and two regions of different concentrations of sugar molecule. Then the two liquids are separated by cellophane paper, which behaves like a semi-permeable membrane. You all know. A semi-permeable membrane is one which allows the passage of molecules selectively. Underline that definition. It can come in exam also. Please underline this. It allows a solvent to pass through it freely but prevents the passage of the solute. This you all know very well. It allows the solvent, that means the water molecule, and prevents the solute passage. Solute means it may be salt, sugar, or maybe sand particles, anything. Now, okay, have you all understood this much?
Yes, ma'am.
Yeah, this was same thing which we studied in the experiment 2 explaining osmosis. Same thing was there using different methods, like different things were used. First rubber sheet was used, then muslin cloth was tried, then we used whisking bath. So by using these 2 to 3 things we were experimenting the process of osmosis. Now we have the topic, how long can osmosis continue? How long can osmosis continue? So osmosis can only continue up to when both the levels are equal, like when in the beaker and in the thistle funnel, when the level of the water or the solution is equal, that time, only up to that time, osmosis can continue. You can read here, osmosis should continue until the concentration of water molecules become equal on both sides of the membrane. That means on the side or other side, that means outer side of the cellophane paper or the semi-permeable membrane, and the inner side, that is inside the thistle funnel. The concentration should be equal on both the sides. Then we have, okay, in the experiment shown in figure 4.4, the column of sugar solution in the thistle funnel keeps rising upwards with the influx of water from the beaker, and with it the height and weight of this column of solution would also increase. Such an increase in the height and weight of the rising column reduces further osmosis. A stage will be reached when no osmosis occurs even if the concentration of water molecules is not the same on the two sides of the membrane. In this state of equilibrium, the water molecules from the beaker tend to force upwards through the membrane, but the weight or the pressure from above holds them downwards. Now look at this diagram, schematic representation of osmosis and the nature of a semi-permeable membrane. So here solution B is less dilute, solution A is more dilute. So from this more dilute solution, all the particles are moving to the less dilute area. That means here the water is fewer. So all the more dilute solution water molecules are moving from solution A to B. Now this pink color, you can see a semi-permeable membrane. So here, basically what is happening? Solution A is more dilute. That means here the concentration is higher. So from higher concentration, the water is going to the lower concentration where the solution is less dilute, the solution B. Clear?
Yes, ma'am.
Yes. Now come to page number 42. You have another thing named osmotic pressure. Now what is osmotic pressure? You all know osmosis needs certain amount of influx, right? You have studied it earlier. An influx or a force is needed for the water to move or for the water to move upwards. You can see from the beaker, it is entering the thistle funnel through a semi-permeable membrane. So it needs some force. Okay, gravity is acted here. So from the thistle funnel also, it should remain there. So it needs some force between them. So that force or that pressure is known as osmotic pressure. You can read this. We may say that osmotic pressure is equal to the weight or pressure required to nullify osmosis. That means the weight or the pressure which we are requiring to nullify the process, to nullify the whole process. Nullify means to balance the whole process, requires some pressure and weight. So that pressure is known as osmotic pressure. I told you from the beaker, water should get inside the thistle funnel through the semi-permeable membrane. Some force is acted here, and that force or pressure or weight is termed as osmotic pressure. Now, here is the proper definition given in this peach color box. Osmotic pressure is the minimum pressure that must be exerted to prevent the passage of the pure solvent into the solution when the two are separated by a semi-permeable membrane. Or very simply we can say osmotic pressure of a solution is the measure of its tendency to take in water by osmosis. So this was what we studied. Taking in of water. Taking in of water by the beaker to the crystal funnel. We have experimented this and we concluded this in this manner that it is the measure of its tendency to take in water by the process of osmosis. You all know what is osmosis? What is osmosis? Can I ask any one of you? Do you know? It is the movement of water molecules from higher concentration to that of a lower concentration through a semi-permeable membrane. You know osmosis? And now what is osmotic pressure? It is the measure of its tendency to take in water by the process of osmosis. That's it. So underline this second definition of osmotic pressure. That is very easy to understand as compared to the first one. So whatever you feel comfortable or you feel that you can understand the definition, so you can write any of these in the exam. Then we have the next topic, tonicity. Tonicity. Relative concentration of the solutions that determine the direction and extent of diffusion is called tonicity. Based on it, the solution can be of three types: isotonic, hypotonic, and hypertonic solution. That means the relative concentration of the solution that determine the direction and extent of diffusion. That means here, tonicity means the concentration of the particular substance which we are taking. It should determine the direction and the extent of diffusion. Concentration of the solution which we are taking, it should determine the direction. That means extent of diffusion means sometimes the water moves outwards from the cell. Sometimes it moves inwards to the cell, inside the cell. So that is the direction and the extent of diffusion. You know what is diffusion? What is diffusion? It is similar to osmosis itself. Diffusion is the process by which the water molecules, that means the free movement of molecules or substance, any substance, from a higher concentration to a lower concentration when they are in direct contact. That should be written in the definition. When they are in direct contact, they are not separated by a semi-permeable membrane. That is the difference between osmosis and diffusion. So diffusion is the free movement of water from higher to lower concentration when they are in direct contact. So, extent of diffusion. That means water moving from higher concentration to lower concentration in a direct contact. So that means the movement of water inside the cell to outside or from outside to inside. Okay, that is referred as tonicity. So you can read this, you can underline this definition: relative concentration of the solutions that determine the direction and extent of diffusion is called tonicity. Then we have three types of tonicity: isotonic, hypertonic, and hypotonic and hypertonic solution. Now, iso means same, hypo means lower, and hyper means higher. Isotonic, tension concentration. The relative concentration of water molecules and the solute on either side of the cell membrane is the same. In such a solution, there is no movement of water molecules across the cell membrane. Yeah. So what is an isotonic solution? Here, in the solution, what happens? Both sides. Solution is present on both the sides, outside also, inside also. But what happens? No passage of water is taking place. Okay, no water is coming inside, no water is going outside. So no passage of water is taking place, or we can say diffusion or osmosis is not taking place. So we can say that no osmosis is taking place in a isotonic solution. That is, iso means same. So same means you could write that it remains the same. The water solution, or sorry, the water concentration in the outside and the inside remains the same. No osmosis is taking place. Clear? Is it clear?
Yes, ma'am.
Yes. Then we have hypotonic. Hypo means lower. That means the solution from the outside is filling the cell, is coming inside. Like we can say, just a minute. Yeah, correct. So the solution from outside is inside, moving inside the cell, and it is making the cell to swell up. Okay, it is just putting efforts to the in the growth of the cell. It is just making the cell to swell up, and that is known as that it takes place in hypotonic solution. And this process, that means entering of water like from outside to inside a cell, it is known as endosmosis, where the cell is swelling itself. And hypertonic means higher. Here, the water from the cell is gone out or is released out, and the cell starts shrinking. So this is known as exosmosis. Clear? So to understand these three, we may suppose that red blood cell and a plant cell have been placed in three different kinds of solutions. So the results may be as follows. Cell shape and size remain unchanged. What will happen? If the cell shape and size is unchanged, the solution outside is isotonic. That means no osmosis is taking place here. A, I'm talking about the A point. Results may be as follows. A, cell shape and size remain unchanged. The solution outside is isotonic. That is same or similar. So that means no osmosis have taken place. Then case B, if a cell shrinks in size and loses shape, the solution outside is hypertonic. That means what is happening here? The cell is shrinking in size. What type of osmosis is acting here? X-osmosis. Okay, you can write it also, X-osmosis. Then case C, cell slightly enlarges or even bursts. That means cell is swelling up here, or it may even burst by swelling. The solution outside is hypotonic. Hypo means here cell is bursting, it is swelling up. So that means water is getting inside the cell. That process is known as endosmosis. Is it clear?
Yes, ma'am.
Okay. Then come to the last paragraph. In the case of plant cells, another striking feature determines the behavior of the cell when subjected to varying external fluid environments. This feature is the rigidity of the cell wall, which resists bulging and protects the delicate cellular parts inside. The phenomena related to this behavior are turgidity, plasmolysis, and flaccidity. Now, in plant cells, plant cells have a cell wall, and inside that, they also have a cell membrane. So the cell wall, it is very rigid and turgid. Rigid means it cannot be bursted easily, or it cannot be swelled up easily with an external pressure or with the help of water. Okay, it is very rigid. It is very firm in its position. So in plant cells, basically rigidity of cell wall is more. That's why they do not bulge or they do not burst out or swell up like in animal cells. They do not burst out. Okay. So they protect the cellular parts inside through that rigid cell wall. So we have three more terms: turgidity, plasmolysis, and flaccidity. We will read it later. Before that, we have 4.4.4 active transport. Active transport. It's the passage of a substance lower to higher concentration, okay, from its lower to higher concentration through a living cell membrane using energy from the cell. So active transport means, okay, we studied diffusion and osmosis. There, the water molecules were moving from higher concentration to lower concentration. In the case of diffusion, there was direct contact. In the case of osmosis, higher to lower concentration with a semi-permeable membrane. But here, in active transport, the passage is just opposite. It is from lower to higher concentration and through a living cell membrane. That means a cell membrane uses its energy to push the water upward. So here the water is being pushed by the cell membrane. So that pushing needs energy. So this energy usage, the process where energy is used, is known as active transport, right? And similarly, there are some processes where energy is not used that much, like like diffusion and osmosis. There, much energy is not needed. They will be known as passive transport. Okay, so here active means energy is required. You can underline active and write energy is required. So when the cell membrane pushes the water upwards, energy is required, energy is created, and thus this process, this transport from lower to higher concentration is known as active transport. Then come to page number 43, phase of an animal cell, RBC and plant cell when placed in three different kinds of salt solution. You can see RBC placed in water. Water entering the cell equals the water leaving it. So when the water entering is equal to the water leaving outside, cell remains, shape remains unchanged because the water amount is balanced. Okay, it is not more or less. So it is entering and it is releasing in an equal amount. That's why cell shape remain unchanged. Then the second case, water entering the cell is very less and leaving is more. So when the amount of water from inside the cell leaves the cell, what happens? The cell starts shrinking. Shrinkage occur. And when shrinkage occur, it is known as exosmosis. So here, this process, the second B portion, it is showing exosmosis. The water leaving is more in amount. Then third one, water entering is more and leaving is less. So when more water is entered as compared to the leaving water, what will happen? It is endosmosis. More water is being taken inside the cell and the cell will burst out. It will swell out. You can see this RBC has bursted out. Okay. Is this much clear to you all?
Yes.
Yes,
Yes.
ma'am.
Okay. Then come to plant cell. Water entering and leaving is equal. So no loss or gain of water by the cell. Okay, it is very equal. Then in the second case, B case, water leaving, water comes out of the cell, exosmosis is occurring. So exosmosis is occurring means the cell is shrinking. Okay, you can see the cell has shrinked inside. It has become small. And the third case, same case like RBC, water is more. So the the cell has swelled up. Water inside the cell, endosmosis is occurring. So the cell is swollen up. Okay, it has not bursted in this case, but it is swollen. So you can see isotonic solution, hypertonic solution, and hypotonic solution. This is the diagrammatic representation of the three solutions. Is this much clear to you all?
Yes.
Yes.
Yes, ma'am.
Okay.
Yes, ma'am.
Okay. Then let us read this paragraph, the last one. Certain nutrients such as ions of nitrates, sulfates, potassium, zinc, manganese, etc. cannot pass through the cell membrane of the root cells easily. This is because their concentration is higher. And it is so maintained in order to develop osmotic pressure for absorbing water. In this way, the concentration gradient of these ions is opposite to that of diffusion. In other words, to obtain them, these ions will have to be forcibly carried inward from the region of lower to the region of higher concentration inside, and this requires energy supplied by the cell in the form of ATP. Now, this paragraph talks about the active transport. Energy is required in the form of ATP. Okay, from lower to higher position, water molecules are carried upwards. So here it is, this paragraph is all about active transport, and below that we have passive transport. Passive refers to requiring no input of energy. I told you energy is not required. You can underline that. Energy is not required. There is a free movement of molecules from their high concentration to lower concentration. What is the example of this free movement? Diffusion. Clear. You can write the example diffusion. Okay, there is a free movement. Now come to page number 44. Turgidity and flaccidity. Turgidity and flaccidity. So Every living plant cell, directly or indirectly, is in contact with fluids. The root hairs, in particular, are surrounded by the soil water. All such cells are subjected to osmosis, and the water continues to enter as long as the cell sap is more concentrated than the surrounding fluids. When a cell reaches a state where it cannot accommodate any more water, it is fully distended, and it is called turgid, and the condition is called turgidity. Okay, so what is turgidity? Just I'm explaining it in a simple way. Sometimes the cell acquires more water or it absorbs more water. The cell sap inside is already high, it is already concentrated solution. So that's why to balance that sap concentration, water is absorbed more by the cell. So at some point of time, what happens? More water if it is absorbing, what happens? The cell gets swelled up or it gets fully filled. It swells up, and because of the swelling up, it cannot continue to absorb water more. So that stage where it cannot continue to absorb the water more, like it has already absorbed so much of water and it has swollen up, so now it is not able to absorb more water. That condition is known as turgidity. Clear? Shall I repeat? Is this much clear? What is turgidity? Everyone?
Ma'am, please repeat one time.
Yes, I will repeat. Turgidity. Turgidity refers to the condition. Okay, sometimes cells acquire more water. You all know, in plant cells and animal cell, we have cell sap. Cell sap is the amount, or we can say some nutrient mediums, and cell sap is basically a fluid which contains all the nutrients and the compounds, etc. So in plant cells also, cell sap is more inside. It is concentrated. It is more. So it should be diluted. So for the dilution, the plant cell is continuously absorbing water from outside. Now, when this water is more inside the cell, like continuously it is absorbing water for dilution purpose, at some point of time it is not able to absorb more water because of the absorption of water molecules from outside. The cell has already swollen up. Now it is ready to burst. Now if it will absorb more water, it will burst out. So at some point of time, plant cells, I already told you, their wall is very rigid. Okay, they have a rigid cell wall. So they stop themselves from bursting out. At some point of time, they do not continue to absorb water. They stop absorbing water from outside. And that condition, when they stop this absorption of water, that condition is known as turgidity. Is it clear now?
Yes, ma'am.
Yes.
Yes, ma'am.
Then, next paragraph. When a cell is turgid, its wall is stretched under pressure from inside, and in its turn, it presses the cell contents towards the center of the cell. The pressure of the cell contents on the cell wall is called turgor pressure, and the pressure exerted by the cell wall on the cell content is called wall pressure. And when the cell is turgid condition means now it is not able to absorb more water from outside. It has swollen up. So because of this swelling up of the cell, the contents which are on the side of the cell, they are pushed inwards, the middle of the cell. That is known as the turgor pressure. Why it is known as the turgor pressure? Because this pressure is created in the turgidity condition. Then some pressure is exerted by the cell wall on the cell contents. Like the cell wall exerts pressure on the cell contents inside. That is known as wall pressure. The wall exerts some pressure on the contents. That is known as wall pressure. And when the contents of the cell, pressure is exerted on the cell contents of the cell wall, it is because of turgidity, it is known as turgor pressure. Then, if at any time the cell wall is unable to bear the turgor pressure, it ruptures and the cell contents burst out. This is exactly what happens when fruits and vegetables sometimes burst. Okay, you have seen vegetables and fruits, okay, like in case of tomato. It will sometimes burst out. You must have seen sometimes in our home when we keep it in fridge also. they used to burst. Why? Because sometimes the cell wall is unable to, hold that turgor pressure. It cannot hold that pressure which is created by turgidity, and it tends to burst out. That's why fruits and vegetables like, tomato, we can take the best example as tomato. It bursts out usually because of the turgor pressure. It cannot hold that pressure. When turgid, the cell is in the somewhat balanced state. No more water is entering or leaving. Turgor pressure counterbalances the wall pressure, and therefore there is no further absorption of water. Even through the concentration of solution inside the cell may be greater than the outside the cell. Okay. Now, when the concentration inside the cell is more, basically what happens, they tend to absorb water from outside. But if the cell has attained turgidity, now it is not able to, absorb water from outside, it will stop there. Okay. It is in a balanced state now. Now, otherwise, if it is absorbing water more and more, what will happen? The cell will burst. So that's why when attaining turgidity, the cell is in a balanced state. Now it will not allow or absorb water molecules from outside. Okay. So you can see this diagram, plant cell showing osmotic pressure, turgor pressure, and pressure represented together, wall pressure. So this is just a diagram. Nucleus is shown, turgor pressure is exerted on the outside, that means on the wall side. Osmotic pressure is inserted or is exerted inside. That means all the organs which are present on the cell wall in the cell wall organs are present. They are exerted inside, and turgor pressure vacuolus and yes, wall pressure also, it is exerting pressure on the cell organs which are present in the walls. The turgor pressure is outside and osmotic and wall pressure is exerting inside. Clear?
Yes.
Okay. I hope this
Yes,
was,
ma'am.
yeah. Okay.
Yes, ma'am.
Okay. Then we have the next topic, plasmolysis and flaccidity. Plasmolysis and flaccidity. When a living cell, such as the cell of a leaf of an aquatic plant, is placed in fresh water, it remains in a fully distended condition. Its plasma membrane remains in close contact with the cell wall and presses it against it, just like a rubber bladder of football pushing against the leather casing. If this plant cell is now kept in 5% salt solution for a few minutes, it will lose its distended appearance. The cytoplasm will shrink and the plasma membrane will withdraw from the cell wall. The shrinkage from the cell wall is called plasmolysis. So plasmolysis is nothing, cell wall, when it is absorbing water more and more, it is swollen up. At some cases, what happens if the plant cell, it is kept in salt solution. Now salt tends the cell to shrink. So when we are keeping the cell in the salt solution for just 5 to 10 minutes, what will happen? The cell will start shrinking. It will withdraw all the water, it will shrink, and this shrinkage of the cell wall is known as plasmolysis. And the cells in the state are said to be limp or flaccid. The condition is known as flaccidity. That means flaccidity is the reverse of turgidity. What was turgidity? Turgidity was the condition where the cell stops absorbing water because it has swollen up already. So reverse of turgidity means, here the flaccidity means the cell is already very shrinked. It has shrinked very much. So it needs water now. It is ready for the absorption of water. So that is known as flaccidity. Here, if however a plasmolyzed cell is returned to water before it is dead, its protoplasm again swells up, pressing tight against the cell wall. The recovery or the reversal of plasmolysis is deplasmolysis. You can easily understand that plasmolysis is the result of outflow of water from the cell, and deplasmolysis is the re-entry. So, have you all understood this much? This is just a reverse thing. Plasmolysis, it is the shrinkage of the cell. Deplasmolysis is the again regaining the shape, the normal shape with water of the cell. The normal shape of the cell is deplasmolysis. Then turgidity is when the cell stops absorbing water because it is already swollen up. And then flaccidity is just reverse to turgidity. That means the cell is very shrinked right now. It is ready to absorb water from outside. Is this much clear?
Yes,
Yes, ma'am.
ma'am.
Okay. So Now we have the terms defined. Turgidity, okay, you have all the definitions here. You don't have to look anywhere. You can underline this much. Turgidity is the state of a cell in which the cell wall is rigid and stretched by an increase in the volume of vacuoles due to the absorption of water. The cell is then said to be turgid. So here, what process is acting? Endosmosis. Okay, water is getting inside and making the cell swollen. Then plasmolysis is the contraction of cytoplasm from the cell wall caused due to the withdrawal of water when placed in a strong hypotonic solution. Here what osmosis is taking place? Exosmosis. Why? The water is going out. The cell is shrinking. So this is exosmosis. Then flaccidity is a condition in which cell content is shrunken and the cell is no more tight. The cell is then said to be flaccid. Okay. These are the definitions for the terms. So underline these definitions, try to learn these definitions. Now we will do the next topics in the next class. Before that, I will tell you something. In your exam, note it down what is the syllabus for your exam. Note it down in your book or notebook. And I hope you are completing your notebook. Uh, write all the question answers up to chapter number 3 and 4 we are doing. We have not completed yet, but I'll be posting the answers of 4th also. If you get in Moodle previous year's answer, please write that also, because 2, 3 and 4, these chapter will be coming in FA1. Chapter number 2, 3 and 4. Structure of chromosomes, genetics and absorption by roots. This will be coming. Also along with that, cell will be coming. Okay, cell, the first chapter, but it will come in a limited quantity. Like you have already studied cell in 9th standard. So I will just put 2 to 3 questions from the chapter cell. Otherwise, more weightage is for the 2, 3 and 4 chapters. Is it clear to you all?
Yes, ma'am.
Yes,
Everyone?
ma'am.
Yeah, have you written? Please write it down. Then afterwards, don't ask me what is the syllabus. I'm telling you right now. Okay, the exam is near. It will be conducted in the month of August itself. From 20th, maybe we will start the exams. Please write the syllabus, complete the question answers in your notebook, and bring the notebooks on the particular dates given to you. Your class teacher will inform you when to bring the notebooks. And then I will check up to 4th chapter. 4th chapter, there are answers in the Moodle, I guess. And if it's not there, I will be uploading today itself. Try to write all the short and long answer type. Not everything, only the short and long descriptive type. Only that much. Is it clear?
Okay.
Ma'am, skill types also.
Yes, Mayank?
Skill type questions.
Skill type question you don't need to write in copy. Just you can practice it for your better knowledge, for your better understanding. You can practice it. The skill type, I have also given you some skill type answers. I have given you from the previous chapter. You can practice it, but it is not necessary to write in the copy. You can write it in the copy because skill type is supposed to be written in the copy. You cannot write all the skill type in the book itself. But it is not necessary. You have to only write. Necessary is only the short answer type and the descriptive type. Clear for every chapter.
Okay, ma'am.
Clear?
Okay, ma'am.
Okay. Any doubts?
No doubt, ma'am.
Okay. Then we are stopping here. We will meet in the next class now.